Okay, so I'm starting with thank you looks like really. So how about you here I am. Good afternoon, everyone. So what I'm going to do really, I'm going to change gear. You have heard a lot about alcohol really. So it's time to talk about something else really. And not too long ago, Missouri used to be meth capital. If you remember, we used to be meth capital in 2012. So before I talk about the work really, I'm going to talk about, this was a graduate student in the lab and then postdoc for 2 years. And he left almost 2 years ago, and now he has a GS14 position after doing a short stint at NIH. Now he's at FDA at GS14, so pretty successful guy. So what I'm going to talk about, I'm going to try to convince you, and you all know that HIV is bad, drug abuse is bad, and our lab works on different kind of controlled and illicit drugs, including alcohol, nicotine also, but today I'm going to talk about methamphetamine for the reason that there are quite a few meth labs in the state, and I've been trying to talk at UMKC that we should have a drug addiction center, but it was so happy to hear from Dr. McCarthy really that you guys are organizing a symposium, and thank you very much for invitation, and thanks for the organizing team for making everything very smooth really. So you can see here, this is 2017 data. My timer is running, yes, 2 minutes. So basically, we have approximately 36, 37 million of HIV positive cases in the world. And you see maximum number comes from Africa and Southeast Asia. How HIV progress? This cartoon depicts if person is not getting any treatment. So one gets virus, develop flu-like symptoms. Most of the time, it goes unnoticed. If you are not in high-risk group, it goes unnoticed. And within few weeks, there is a high viral load. Viral is replicating massively. Immune system comes into play, controls the virus, but immune system is never able to get rid of the virus. And virus is replicating predominantly in CD4 T cell, and therefore CD4 T cell number goes down, immune system takes care, not eliminating. So CD4 goes little bit, there is little recovery, but over a period of time, virus is very smart, just next to flu virus in terms of mutation really. So virus keeps mutating and immune system is going down. There comes a time, 3 to 10 years without treatment, that host immune system is totally compromised and patient is going to succumb to some opportunistic infection. What happens in the brain? Today, I'm talking about the brain, really. So what happens in the brain? Virus goes in the brain, cross blood-brain barrier. There are 3 major cell types: astrocyte, microglia, and neurons. Virus does not replicate in neuron, but does replicate in microglia and astrocyte. Our lab is focused on astrocyte. So this slide gives you an idea that what used to happen before we had combined treatment. These days we have great treatment. We cannot get rid of the virus, but we can keep virus in check, and it's more of management issue rather than, so HIV/AIDS is not a death warrant anymore. So before HARD, we used to have approximately 30% people who will develop dementia, severe brain disorders, really. But now that lifespan has increased, so we don't have, and we have good drugs, much better than 90s, so we don't develop dementia. We develop minor cognitive motor dysfunction, so our executive function, memory is, but since we are living, so in nutshell, if one is infected with HIV at the age of, let's say, 25, by the time that person reaches 40, his brain would be as old as 65, 70-year-old brain, really. So brain aging is much faster in case of HIV. So let's talk a little about HIV because I'm going to focus only on one protein of HIV today. There is one protein any virus has envelope. So there is a protein on the envelope that is called gp120, this one. That binds with receptor on the CD4 T cell, and that's how virus gets in. So gp120 is ligand for the viral receptor CD4, and also very important, over a period of time it has been shown that it's extremely neurotoxic. One of the two proteins, gp120 and TAT, they are neurotoxic, and the lab works on both really. So it does all different kind of thing really, and in the literature it was known that it is neurotoxic, and that time I was moving to Kansas City, and I used to be monkey researcher really, but in University of Missouri we don't have monkey facilities, so I tried to diversify, became tissue culture and rodent researcher. So that's how the evolution was. People go on positive side. I went from bigger to smaller really. So that was the time this guy Ankit came to lab, and I gave him this project, and we had a R01 based on this project really. So he was able, and I'm not going to show any data, just going to tell you what happens really. So he published 3 papers. So what happens? How our neuron gets compromised? There are several mechanisms, direct, indirect. Something can go directly hit the neuron. Something can go activate astrocyte, make them do bad things, and those bad things will affect neuron. And one of the bad things is pro-inflammatory cytokine chemokine. So he was able to show that gp120 causes overexpression of IL-6, IL-8, and CCL5. And all these 3 are bad. If they are being overproduced, it will compromise normal physiological function because there will be inflammation. And he was able to develop a signaling mechanism for overproduction of these 3 cytokines, and that is illustrated here. I'll not go into detail, but he was able to beautifully dissect the pathway that was involved in overproduction of these 3 cytokines. Now let's come to methamphetamine. So we know that HIV is doing bad thing. So we chose methamphetamine for particular reason. So while looking at methamphetamine, there is one comprehensive study in 2006 when SAMHSA, in collaboration with NIDA, they found that almost 2 million high schoolers admitted using methamphetamine at least once. So this is countrywide data, and it causes different, amphetamines are good also for ADHD, narcolepsy, and all that, but if one becomes addicted, then there are bad things that happens, memory impairment and all those, and we have a lot of data on that, but we are not going to talk about that. How does it work in context with HIV? So methamphetamine affects dopamine and dopamine transporter, and on the other hand, dopamine metabolites are known to affect HIV/AIDS disease course. So that is kind of thing, and there was a study that dopaminergic drug could affect the SIB neuropathology, that they were able to control severe pathology to milder level. Coming back to Missouri, like 2012, we were meth capital. This data is from 2013, when we were overtaken in a good for us, overtaken by some other states really. But look at this data. This is from Missouri State Highway Patrol. If you look at from 2010 through 17, and there are data on that side, data is there from 2004, but I didn't take that. So you see approximately 10% of the methamphetamine-associated seizures are reported in the state. So it's still a big problem for the state, really. So we were working. So on one hand, we were able to show that HIV protein gp120 causes inflammation in the brain. On the other hand, Ankit went ahead and tested methamphetamine, and he was able to show that IL-6 and IL-8 were overexpressed when astrocytes, in one of the brain cells, were exposed to methamphetamine. And he did all those classical biochemical studies and was able to identify the complete pathway. So since I started working with HIV, substance abuse was the second phase in the life, really. So we always study substance abuse in context with HIV. So methamphetamine, still there is no single study clearly depicting that methamphetamine causes increased HIV/AIDS. So there is no study. But there are two classical studies, and NIDA has awarded a lot of R01 based on that study, really. They were conducted in 2006, where people from UCSF found out that incidence of HIV is 2 to 3-fold higher in those people who uses methamphetamine versus who don't use. So HIV incidence is higher. So that was and since then, people have shown that HIV replication in vitro is increased. We had a monkey project, and there we are not seeing increased HIV replication. We are seeing accelerated disease, not for today, but we are not seeing increased HIV replication. So what he did, Ankit, he mixed now gp120 and methamphetamine. And he was able to show that at least for one cytokine, IL-6, there was synergy, that 2 plus 2 became 11, not 4. So if cells were exposed to gp120 and methamphetamine, there was synergistic effect in terms of IL-6 production, and that's what we have shown now in mouse also, that there is increased memory deficit. So that was inflammatory part. There are two other mechanisms by which brain cells are affected, or any cells are affected, or cells would die. ER stress, endoplasmic reticulum stress. I'm not going to go into detail. This paper was published in OncoTarget. And what he was able to show, that if you expose the cell to methamphetamine and also expose the mice to methamphetamine, they will involve 3 pathways, ATF6, IRE1 alpha, and PERK pathways, and cause increased ER stress that will lead to increased cell death and compromise in brain function in mouse. So both tissue culture and mouse study. Another thing is oxidative stress. We all know that means one of the common, when we are 20, 25, our skin is so smooth. And when I'm teaching and I put my hand under overhead projector, first thing I tell students, hey, look at, this is the aging, old person really talking. So that is because of increased oxidative stress, that as we age, we make more oxygen, reactive oxygen, and that will cause all the abnormalities. You guys are lucky sitting there, but just wait for 2 decades, really you'll be here. So that is classical aging, okay? But if a reactive oxygen is produced, for some reason balance is impaired, then that will cause death. And eventually, one of the reasons for death is aging, death, oxidative stress is major player, really. So means there was a time I used to be very smooth, really, not like this today, what you see me. So oxidative stress is one mechanism. And that's what we did. We published only one paper on that. We took HIV protein and methamphetamine. We showed that there was increased oxidative stress, nothing big. It was already known before we came and performed this study. What was new in our study, that we discovered a new pathway, that cytochrome P452E1 is involved in gp120 and methamphetamine-mediated oxidative stress. So this new pathway was published in Cell Death and Disease few years ago now, 13 or 14, something like that. And we were able to show that these things cause oxidative stress that leads to cell death. And if you mix those two together, there is synergy between these two. There will be increased cell death. And we have not pursued gp120 in the mice, but we have pursued HIV, another neurotoxic protein, HIV-1 TAT, and have shown that's exactly what's happening in the mice, and that is leading to compromised downregulation of neuroplasticity genes, leading to memory deficit. And if you block this, then you can recover memory deficit. Our lab is also working on natural products. So basically, we are fan of broccoli extract, really, sulforaphane. So we test sulforaphane, really. We have few resveratrol analog also. Resveratrol, the wine thing I was talking about, resveratrol is wine thing. So that has its own problem in terms of half-life and bioavailability. So we have designed few analogs. Those are better, but sulforaphane is working much better in terms of restoration of executive function and memory deficit. So I think that's where kind of we are really. And methamphetamine, I chose methamphetamine. I'm sorry. I chose methamphetamine simply because of our problem in the state. But we are pursuing different things like opioid, morphine, heroin, cocaine, alcohol, and cigarette nicotine, and trying to work out what are the mechanisms, how the cells are dying, how you can restore the memory or any executive function. So that's kind of, and like I told you that there were other people also involved in this project, but Ankit, this was Ankit's PhD thesis and postdoctoral work, and funding came from NIDA and NIAAA. Thank you very much.